Most eukaryotic regulation takes place at transcription because this is where the cell's going to save most of its energy. But truly, it can be regulated at any of the levels that we discussed. It can be regulated at DNA structure, it can be related before or after transcription and during transcriptional modifications. It can be regulated before, during, and after translation. And one of the major ways that eukaryotic cells regulate gene expression besides regulation at transcription is by structural changes in the DNA. And for time's sake, we are not going to cover all of the different types of structural modifications. But I do want to talk about a topic called epigenetics because this is one of the newer topics in the field of genetics. And our textbook doesn't really cover it in too much detail. If you are interested, I highly encourage you to do a little bit of research and learn a little bit about some of the examples of epigenetic modifications. So epigenetic modifications are basically structural modifications in the DNA that are inherited. And so we are going to focus on our epigenetic modifications and not so much our other types of DNA modifications here. But I just want you to know that there are some structural changes that can take place to regulate gene expression. You can also regulate alternative splicing by putting together different exons, and then you can also use RNA degradation to control gene expression. For epigenetics, epigenetics is really kind of interesting because we normally think of heritability as genes that are inherited from one parent to the other, and we think about changes in those DNA sequences itself. So we normally think about inheritance as changes in the nucleotide sequence of our DNA. But epigenetics is a little bit different. Epigenetics is the study of the inheritance of changes in gene expression. Inherited changes in gene expression come as factors that affect the phenotype of the individual without changing the DNA sequence. And the reason that this is so interesting is that the sequence stays the same, but whether or not a gene is expressed is passed on to the next generation. So by the nucleotide sequence, someone might have a specific predicted phenotype, but they may actually express something different because of epigenetics. So when you have structural modifications to the DNA that are essentially permanent, these are passed on to daughter cells during cell division. And so all future generations could potentially have a gene turned off that maybe should be turned on, or at least we would expect it to be turned on. So this can come from changes in chromatin structure. It can be caused by environmental factors or inherited. And then because they can be acquired in an environment, from an environment, they can be modified over one's lifetime. So you can actually acquire epigenetic changes. So the epigenome is essentially the pattern of modifications that each individual has. So let's take a look at one example of how an epigenetic modification might take place. In epigenetic methylation, we have methylation of a DNA nucleotide. So a methyl group is a CH3. And very commonly, the cytosine nucleotides are what is methylated. Now, DNA remains methylated through replication. And if you add in a new nucleotide, then the corresponding nucleotide is going to have to be methylated as well. So this step is essentially maintained as if it were a change in sequence. So what happens here is that during DNA replication, the new molecule has not been methylated yet, but a molecule, and I'm not worried if you know about this name, comes through, the methyltransferase enzyme comes through and recognizes where a methyl group has already been added and adds one to the other side. And so now we have a methyl group on both of our sides of our DNA. Now, the thing that's important here is that methyl group changes the structure of the DNA. And when it changes the structure, it can block transcription. So if the RNA polymerase doesn't recognize the DNA sequence, then transcription won't happen for that particular gene. And so if a gene is turned off because of methylation, and methylation maintains itself through replication, then that means that this methylation can be inherited. And so by this methylation, we have an inherited mechanism of transcriptional regulation. Whereas in a cell that is not being regulated by methylation, would just express it based on the environment or the conditions of the cell. There are other structural modifications. I skipped over the structural part, but one of them includes modifications to the histone molecule rather than the DNA molecule. This is going to affect the wrapping and packing of our DNA molecule, which would then in turn affect whether or not that molecule can be transcribed. Epigenetic changes may be the reason that sometimes our monozygotic twins or identical twins are not actually identical. Because methylation can happen during an individual's lifetime, this also means that two individuals might have differences in methylated DNA, which means they are going to potentially have differences in gene expression. So as these environmental effects cause differences in methylation, these twins could potentially grow more and more dissimilar the older they get as they accumulate changes in their gene expression due to epigenetics. There was a study that backed this up, a study of 80 monozygotic twins done in Spain at the National Cancer Center in Spain. And this shows that epigenetics are often similar in early life, and this makes sense if epigenetic changes are passed on from the parent. But as individuals grow up, they become more and more different, suggesting that you can accumulate these differences in gene expression over your lifetime. So these changes can come from a variety of different places. They can be induced by maternal behavior, so something that the mother does to cause a change in epigenetic gene information. It can be a stress response, whether that is a physical stress, an environmental stress, whatever the case may be. We talked about changes to our DNA based on things in the environment like UV and chemical exposure. So they can also cause structural changes in DNA, not just damage, but permanent structural changes. And then we can also have just changes in across generations. So as individuals are affected throughout their lifetime, their patterns of methylation and gene expression based on physical characteristics can change as well.